Hey everyone, in this video we are going to talk about the FRQ practice on cell types. So it says that the following data were collected by observing subcellular structures of different types of eukaryotic cells. Based on an analysis of the data, identify the likely primary function of each cell type and explain how the data support the identification. So cell type X, let's start with that one. And they tell you the relative amounts of the organelles that are in the cell type. So we're going to start with cell type X. You'll notice it has a small amount of smooth ER. It's got a small amount of rough ER. It's got a large amount of mitochondria, and it's got cilia present and small amount of Golgi bodies. Based on the organelles that are present in large amounts, we would safely predict that the function of cellular cell type X is some type of movement or locomotion. The reason we would know this is because, number one, it has cilia present, and cilia are present for movement on the outside of the plasma membrane. The second thing that we also would use as justification for movement or locomotion is the fact that it's got a large amount of mitochondria. A large amount of mitochondria would provide a large amount of energy, which is needed for movement or locomotion. Next, we're going to look at cell type Y. You'll notice that it's got a large amount of smooth ER, a large amount of rough ER, a moderate amount of mitochondria, no cilia, and a large amount of Golgi bodies. So keep in mind that Golgi bodies work hand in hand with the rough ER with the secretion of proteins. It's also got a large amount of the smooth ER, which makes lipids such as phospholipids that would be needed to make vesicles. Based on these observations, we can guess that cell Y functions in secretion, because these are the organelles that would be abundant in cells that do a lot of secreting and creating vesicles that are going to fuse with the plasma membrane to exit out into the extracellular environment. The last cell, cell Z, you'll notice is absent, 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 absent. Has none of the organelles. So you might think that it's a prokaryotic cell. However, in the prompt, they tell you that they are eukaryotic. So either it was mislabeled and is in fact a prokaryotic cell, but the more likely explanation would be that cell Z is a red blood cell. Remember that red blood cells have none of the organelles that are involved with normal function. Red blood cells, what they do is they provide a means of transport of oxygen. So to maximize that amount of space, they have no other organelles inside their plasma membrane. Thanks so much for watching. I hope this video was helpful, folks. Bye.